All right, so we're going to talk about glycolysis, which is the first part, the first stage of the oxidation of glucose. So glycolysis basically means the splitting of glucose. It occurs in the cytoplasm of the cell and is anaerobic. So it does not utilize oxygen at all. So it starts with a molecule of glucose. Glucose is coming into the cell, and glucose is a 6-carbon molecule. And as glucose comes in, the cell changes that glucose to something a little bit different so that the glucose won't leave the cell. So it changes it to a molecule called glucose 6-phosphate. So the carbons on the glucose are numbered 1 through 6. So if this is the 6th carbon, we're going to add a phosphate to it. And that will make the glucose become glucose 6-phosphate. So the phosphate being on that 6th carbon. So where do we get that phosphate? It just doesn't appear. It comes from ATP. So ATP, we take the phosphate off of ATP, it becomes ADP, and we attach it to the glucose, making glucose 6-phosphate. From there, it changes, it gets converted to something called fructose 6-phosphate, and from fructose 6-phosphate, it is converted then to, it's converted to another molecule where we add another phosphate to it. So we end up, during the phase in glycolysis, which is the energy consumption phase, 1, 2, 3, 4, 5, 6, we have phosphate here from the glucose 6-phosphate, and then we transferred that or converted that into fructose 6-phosphate, and then added another phosphate to it. Okay, this molecule is called fructose 1,6-bisphosphate. Because the phosphates are on the 1st and 6th carbon. Okay, so again, we added another phosphate group, and we got that from ATP. So we used another ATP molecule, took the phosphate off of it, it became ADP, and we added it to this molecule, these 6 carbons, producing fructose 1,6-bisphosphate. Once we get to here, this molecule is cleaved. So it's cut in half here, and it becomes 2 3-carbon molecules. Carbon, 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 carbon. And each one of them has a phosphate on it. So if this is the 1, 2, 3 carbon, this is the 1, 2, 3 carbon, this molecule here is called glyceraldehyde, 3-phosphate. Because the phosphate is on the 3rd carbon. This one over here is called dihydroxyacetone phosphate. Dihydroxyacetone phosphate. So I'll just put that like that. And it really cannot go further in this glycolysis pathway. So this molecule here will be converted into glyceraldehyde 3-phosphate. So we'll end up with 2 of these molecules. From there, we're basically, and let me make this line a little bit longer. So from here, we are going to convert this glyceraldehyde 3-phosphate into a 3-carbon molecule called pyruvate. Okay. We go through, there are 3 or 4 steps in here that cause that, and we're not going to go through that, but we need to be aware of what we get when we're doing these reactions here. And the first thing we get in this reaction is an NADH. So we have an NADH plus, and we get an NADH there. And that comes from converting the glyceraldehyde 3-phosphate into another molecule, which gets an inorganic phosphate added to it, and then the NAD takes up the hydrides. Okay, so we end up with an NADH, which is an electron carrier. So we have 2 electrons now with this NADH. Now, we have 2 NADHs because there are 2 glyceraldehyde 3-phosphates. So we end up with 2 NAD. There's 2 NADs, and we end up with 2 NADHs. Okay. We also, within this, we use the phosphates off of these 2 molecules, and we're going to get ADP. We're going to add the phosphate from these molecules to ADP and get ATP. ADP to ATP. That happens twice, and there's, again, 2 molecules of this happening, or it's happening 2 times, so we're getting 2 here and 2 here, 2 ATPs here. Okay. So this process right here, this ADP to ATP, is called substrate level phosphorylation. So it's just the direct adding of the phosphate group back to ATP. And we do that twice here. So in the end, we end up with this 3-carbon molecule. The phosphates are gone because they're attached to ATP, and we end up with this 3-carbon molecule called pyruvate. Pyruvate has 2 things that can happen to it. So one thing that can happen to pyruvate is if there's no oxygen available, or we're doing something that there just isn't oxygen to be had to use, it will go through fermentation. And we will get lactic acid. Lactic acid will go to the liver, and it can be converted back to glucose. The liver can make ATP out of it, whatever. Okay, so there's lactic acid. This is the fermentation process. Okay. The other thing that can happen is if there is oxygen, it can enter the mitochondria. And if it enters the mitochondria, then it can be converted into a molecule that will enter something called the Krebs cycle. And that will continue the breakdown, the oxidation of that glucose that we started with. Okay, so the final final total for glycolysis is we get 2 net ATPs and 2 NADHs, which contain each one containing 2 high energy electrons. Okay, so these things can be used later. These high energy electrons can be cashed in later for more ATP. Now, this number, 2 net ATP, don't forget we do make 4 ATP here, but we use 2 up here in the beginning of glycolysis. There was a phase, this phase here, which consumes energy, and we had to use ATP to get those phosphate groups to continue it along. So the net, again, the net products out of glycolysis are 2 net ATP and 2 NADH, and also 2 pyruvate. And those 2 pyruvate will go into the mitochondria if there's oxygen available, and it'll be converted further to continue the oxidation of this molecule glucose, or it can go to basically be converted to lactic acid through the fermentation process, go to the liver, and do some other things there. So the next video, we'll talk about the next step, the Krebs cycle.